Welcome to the unit on sex chromosomes and gene linkage. As we have discussed previously, the homologous chromosomes in each cell are a pair of chromosomes that match in size and shape and have the same genes in the same order. However, in many species, there are a pair of unmatched or non-homologous chromosomes called the sex chromosomes, which differ between different sexes in the species. In humans, the sex chromosomes are called the X chromosome and the Y chromosome. In humans, the male is XY, while the female is XX. You should note that not every species uses an X and Y chromosome for sex differentiation. Some animals, such as birds, have a ZW sex determination system, where the male has two Z chromosomes and the female has a Z and a W. Others, such as insects, have an X chromosome but no Y. The presence of one X determines male, while two X's are female. Some organisms have no sex chromosomes at all. For example, in bees, haploid cells develop as males and diploid cells as females. In others, such as some reptiles, sex is determined by the temperature that the eggs are hatched at. When we look at the human sex chromosomes, the X chromosome is more than 150 megabases long. This means that there are 150 million A's, T's, G's, and C's that make up the chromosome, while the Y chromosome is only about 50 megabases. The X chromosome contains more than 1,000 genes, and the Y contains only about 50 genes. While the chromosomes themselves are very different, there's a small region of sequence homology, DNA sequence that has the same spelling of nucleotides, found at each end of the chromosomes. This small region is necessary for alignment of the chromosomes in meiosis, because it is how the sex chromosomes form a tetrad during prophase 1 of meiosis, even though they are not homologous chromosomes. When gametes are formed during meiosis, the sex chromosomes separate from each other similarly to all the other autosomes in the cell. Because females are XX, they only have an X chromosome to give to the next generation. So all eggs will contain a single X chromosome. Human males are XY, so the X chromosome will separate from the Y chromosome during anaphase 1 of meiosis, resulting in half the sperm bearing an X chromosome and the other half bearing a Y chromosome. With random fertilization, half of the fertilized eggs are expected to be chromosomally XX, while the other half is XY, which is characteristic of Mendelian segregation. Interestingly, this ratio refers to the sex ratio at the time of conception. However, we see a slightly different ratio at birth, with a slight excess of males. In the United States, this ratio is approximately 100 females to every 105 males born in the country. X-linked genes are genes that are found on the X chromosome. It is important to know if a gene is X-linked because it will display a different inheritance pattern between males and females than genes on autosomes do. This is due to the fact that females will have two copies of the gene, similar to all the other autosomes, but males will only have one copy because they only have one X chromosome. This means that females can be homozygous dominant, homozygous recessive, or heterozygous. But males are what we call hemizygous, meaning they only have one copy of the gene. They will display either the dominant or recessive phenotype depending on the allele they have. X-linked recessive traits appear more often in males than females because they are unable to hide the recessive allele in a heterozygous genotype. You should also notice that the allele of the genes is written as a superscript on the X chromosome. This is to remind us that they are alleles of an X-linked gene. An example of an X-linked recessive mutation in humans is red-green color blindness, which affects approximately 1 in 20 males. Color blindness is the recessive allele, while normal vision is the dominant. Individuals who are colorblind will have trouble seeing the numbers 97 in this picture. We will walk through an X-linked genetic problem to demonstrate the importance of including the X and Y chromosomes when completing an X-linked cross. In this problem, a colorblind man will have an X and a Y chromosome because he is a man, and his X chromosome will contain a recessive colorblind allele. The female must be XX because she's female and she has normal vision, so at least one of her X's must have a dominant allele. However, she could be homozygous or heterozygous. Since the problem tells us that her dad was colorblind, she must be heterozygous. Since the X chromosome would have come from dad, so it must have the colorblind allele. We will then set up our Punnett square, keeping track of both the X and Y chromosomes. The sperm could have an X with the colorblind allele or a Y, whereas the eggs could have an X with the normal vision allele or an X with the colorblind allele. At fertilization, we see these combinations which could come together. Now we will answer the question. The question asks, what is the chance that this couple will have a colorblind son? In this case, we will need to look at both sex, we're only looking at males if we care about the sons, and colorblindness. So what that means is out of 4 children, they will only have a 1/4 chance of having an offspring who is both colorblind and a son. However, if the question was worded in a different way, such as this, where it says the couple has a son, what are the chances he is colorblind, then the answer would be a 1/2 chance. This is because they already had the son, so we already know that the boxes with daughters cannot be options. This means there are only 2 options for the son. In one option he's colorblind, the other he is not. Thus a 1/2 chance of being colorblind. When examining human pedigrees, you can often identify an X-linked recessive inheritance pattern by looking for key features. These include that the affected individuals are almost always males, because males only need 1 copy of the mutant gene to be affected, whereas females need 2 copies. Additionally, affected males will have unaffected sons because males transmit their X chromosome only to their daughters. A female whose father is affected can have affected sons because she will be a heterozygous carrier of the recessive mutant allele. One of the most famous examples of a human X-linked inheritance is the trait of hemophilia, which results from a recessive mutation in a gene encoding a protein necessary for blood clotting. Affected individuals bleed excessively even from minor cuts and bruises. Hemophilia is famous because of its presence in many members of the European royalty who descended from Queen Victoria of England, who was a heterozygous carrier of the gene. You will notice the double lines throughout this pedigree due to in-family marriages that occur because they're trying to marry only without royalty.